Hello, everybody. Thank you so much for joining us today. My name is Emma Whiteman, and I'm a senior content marketer and tech writer here at Triumvirate Environmental. I'm so excited to be your moderator for today's Service Spotlight. These Service Spotlights are a bit different than our webinars. You'll hear a quick presentation from our expert before we turn it over to a lengthy Q&A session, and we'll wrap up at 2:45. Our informative long-form webinars will still be offered throughout the year. But before we get started, I'd like to share that everyone's mics and cameras will be turned off for the entirety of the webinar. You can use the questions tab on the right side of your screen to communicate with us and ask questions. Please keep in mind that you will receive a copy of the slide deck and the recording in the days following the event conclusion. If your question goes unanswered, you can follow up with us. Today, we continue our Service Spotlight series with biosafety, and I'm so pleased to introduce today's speaker, Dr. Brandon Linz. Brandon Linz, PhD, RBP, is Triumvirate Environmental's EHS Director of Biosafety. He's a registered biosafety professional and holds a PhD in microbiology and immunology, with a concentration in infectious agents and high containment facilities. Dr. Linz has over a decade of biological safety, biologics manufacturing, research and development, and higher education experience, and manages a skilled team of delivery consultants at Triumvirate Environmental. So without further ado, I'm pleased to turn it over to Dr. Linz.
Thank you so much for that introduction, and thank you everyone for coming today. I'm pleased to talk about biosafety and the services that Triumvirate can offer. So what is biosafety? It's the discipline within environmental health and safety focused on mitigating the risks of exposure to or release of biologics. These can be infectious agents, biological toxins, genetically modified organisms, ants, animals, plants, and anything in between. The fundamental core of a biosafety professional and our engagement with an institution or a lab process is fundamentally the risk assessment. During this process, we identify any agents, evaluate their pathogenicity, how they are transmitted, what the host ranges are, and any availability of treatment prophylaxis. We look at actually what lab procedures are going to be done, assess those activities that might increase exposure risk, either through aerosolization of the material, sharps that could lead to a cut or a percutaneous injury, and the scale, the volume of what materials we're dealing with. We assess the personnel, the facilities, considering how the staff are trained, if there's immune status available, and the physical containment capabilities of the facility or either the engineering controls. We assign appropriate containment levels. This is directly through the biosafety levels 1 through 4, and any necessary controls, including engineering, personal protective equipment like gloves and lab coats, and any administrative standard operating procedures. This risk assessment also evaluates the recombinant synthetic processes and modifications, ultimately ensuring compliance, namely with the NIH guidelines and the Institutional Biosafety Committee engagement. And then last is implementing the mitigations and oversight, either through document controls, the emergency plans, updating the risk assessments, and then training the staff on any changes in how to safely operate within their own facility. Fundamentally, most of the biosafety professionals' work revolves around the National Institute of Health and the IBC, the Institutional Biosafety Committee. So the NIH guidelines for research involving recombinant or synthetic nucleic acids. It's required for institutions who have NIH funding and some other federal funding, but it is a best practice for the risk assessment process and the review process of recombinant DNA research is inherent to the Institutional Biosafety Committee's charge. So through this, as a documented formal way, the IBC, composed of various members of recombinant DNA experts, infectious disease experts, the animal plant experts, depending on what work is being done, biosafety professionals, and then non-affiliated community members all bring to the table their combined expertise to look at the risk assessment we talked about on the last slide and how to safely implement that work at their institution, confirming that the facility is adequate for the work occurring, confirming that the staff have the appropriate trainings, and then all the waste is not going to get out and be released into the community. So recent updates that are happening within the NIH and the IBC reporting process is that for all registered IBCs, so this is all with NIH funding or otherwise required to comply, the meeting minutes for these IBCs must be made publicly available starting at the beginning of this month. So what that means is that once those IBC minutes have gone through a review of the committee, likely at the subsequent meeting, and then reviewed by the committee and approved, those minutes then have to be publicly posted on the company website. Additionally, as part of these updates, the NIH has made the following information for the registered committees publicly available, including the IBC chair, the IBC contact, as well as the biosafety officer for the institution. So when we're looking at our risk assessments, oftentimes there's a large disparity between R&D and biomanufacturing. One of my previous webinars was in biomanufacturing and the risk assessments inherent to this. So if you're curious for a more in-depth dive, please encourage you to go watch that. But when we're looking at small-scale research and development, these are very few small processes, often limited to small milliliters up to a few liters. Whereas large-scale biomanufacturing can be to tens, thousands, or even tens of thousands of liters for a production vessel for certain pharmaceutical products. We're focused on flexibility and discovery versus a large-scale biomanufacturing is all about the FDA-mandated consistency and containment for the product safety. When we're looking at the research scale, often we're concerned with primary barriers, so our biosafety cabinets and our PPE, but in the large scale, we're looking at closed systems and facility design, ultimately to maintain there are no releases to expose any research, production personnel, or the community. Regulations vary, so within the small scale R&D, it's heavily focused on NIH and CDC. For large scale, it's the FDA, EPA, and often global regulatory compliance because they are producing regulated pharmaceuticals. Documentation varies from basic research SOPs all the way to large-scale validated SOPs with global manufacturing, good manufacturing processes. And personnel, it varies from basic research focus and lab academics to good manufacturing processes, industrial hygiene, and large-scale spill emergency responses. BSL-2 plus and BSL-2 enhancements is also something we work with a lot. This is BSL-2 plus is not a defined biosafety level within the CDC's BMBL, but rather it's a BSL-2 facility utilizing BSL-3 practices and procedures. So this is all dictated by the risk assessment and looking at what additional processes could be mitigated to reduce the risk of exposure to personnel. So for aerosolizing agents, we often look at additional PPE and general engineering controls to address these risks. So materials that are routinely handled at BSL-2 plus could be viral vectors, most commonly lentiviruses that are pseudotyped for humans and that are a certain generation where we have inherent risks to the pseudotyping itself. And especially if we have toxic or oncogenic inserts that may cause the viral vector to transform the human if they were to be exposed to it. Non-human primate materials are also of note at BSL-2+, especially for primary tissue samples. There are certain zoonoses that non-human primates can carry, namely herpes B. And so BSL-2+ in this case would be additional PPE, potentially awareness training, and then occupational health exposure prophylaxis if an exposure were to occur. But all this is inherent to the risk assessment, the procedural development, and then training of personnel working in these labs. Triumvirate does a lot of work as well as in our BSL-3 and animal biosafety level 3 startups. So we help do permitting and regulatory review from basic CDC and USDA engagement all the way up through select agent registrations. Program and SOP development from the BSL-3 manuals, the basic risk assessments for the IBC, and then procedural template developments. We also work with the lab operations team and the laboratory research team to design special trainings and drills for personnel onboarding, emergency responses, annual refreshers, as well as town primary emergency response resources doing live exercises or tabletop drills. We also work with facility design and review. So we work with construction companies during the outlay of large facility manufacturing, construction to help lay out the consultation, workflow optimization, and containment verification requirements. We support commissioning and certification processes for these large scale facilities. And then we help with inspection readiness and audits for initial startup work as well as ongoing triennial inspections from the USDA or CBC as they come up. So as I mentioned, we work with a lot of local, state, and federal institutions for waste disposal requirements, local RD&A biosafety permitting, CDC imports, USDA APHIS for both plant and animal health agents, U.S. Fish and Wildlife for endangered species, including most non-human primates, and then shipping and transport compliance therein. I do want to note, additionally, there is some biosecurity executive orders that is impacting the biosafety community currently. So for the dual use research of concern, or DIRC, and then pathogens with enhanced pandemic potential, or PEP. So these policies were promulgated and were to go into effect earlier this year, but a recent Trump administration executive order issued earlier last month overrides this by NERA policy and gives 120 days to the NIH to revise this policy. So in the Biden era administration, this was only to apply to institutions receiving federal funding, but the new Trump administration rules have already indicated that this will apply to those without NIH or federal funding. So many programs will need to update their biosafety processes, how their IBC reviews work on site to ensure that it does not go into the world of dual use research of concern in which bioterrorist agents could be created at this site. So a lot of processes if you're working with these agents, and additionally you'll have to do vendor verification of any nucleic acid vendors like PCR primers, or if you're working with a third-party DNA cloning institution to perform work, there has to be verifications of these that they're not going to send you any select agent materials. So Triumvirate Biosafety Services, we offer anything from a single off project all the way to ongoing maintenance and program management. So we perform gap assessments, risk assessments for your new work, IBC development and support. We can help local, state, federal permitting for both import and export, as well as use permits. We can help with your training, all the way from basic biosafety trainings, the 2+ trainings, 3, and then any agent-specific trainings. We help with biosecurity, as I mentioned before, both with BSL-3 startups as well as DIRC and PEP compliance. And we can support your staff through basic project work, or all the way up through staff augmentation with a biosafety professional working on site with you and your team. And lastly, the SOP and biosafety manual creation if you're starting a new project or a new site. So with that, I want to thank you all for your time. I really appreciate you the service today. And with that, I'm going to throw it back to Emma.
Yeah, thanks, Brandon. What an incredibly informative presentation. I know I definitely learned a lot about biosafety and all the different considerations that they have. One thing that someone was already curious about is what exactly would using Triumvirate Environmental for biosafety services look like from start to finish, from contacting you to having a successful biosafety program? Can you walk us through that a little bit?
Absolutely. So I'm going to give you the classic biosafety answer of it depends. Every institution is going to be different in what the scope of their biosafety work is, let alone how it is going to impact them. So we would meet with you and your operations or safety team, depending, understand what scope of work your institution works with, and then we'd recommend actions of how to go from there. Either performing a gap assessment to understand where your program is and recommend any updates to it, especially with the go forward model of how Triumvirate and our biosafety team can help you support those go forward goals.
Yeah, that's a great point. And how do I need biosafety services even if I don't have a assigned biosafety level?
It's an interesting question. So if you're not inherently working with biologics in a research or manufacturing, so if there's no biosafety level, the answer is unlikely. But many institutions might have a first aid program where their members are working with potential blood and bloodborne pathogens. So they would indeed need a bloodborne pathogens program, which falls well within the biosafety realm of what Triumvirate can offer.
Sorry, I accidentally turned my camera off instead of turning my mic back on. That's great. And someone here is asking about you mentioned some federal biosafety considerations for people to take into consideration. Do state and federal biosafety regulations differ, or will they be largely the same?
So Again, it depends. So as far as the federal, those are very guidelines that apply regardless if you have applicable funding sources. But as far as superseding regulations, they don't tend to differ. What will differ is the state to state variation on waste regulations and then other programmatic requirements.
And is there one central location or resource where people can have access to these requirements?
Unfortunately, no way to do it outside of biosafety practicing as a biosafety professional and understanding where the nuance applies.
Sure, that makes sense. Someone here is asking, they have multiple potential biosafety risks in their facility. Do they need a separate risk assessment for every single one of those biological or infectious agents, or would one risk assessment cover all the areas of concern?
That's going to very much depend on the institution as well. So for a single site, so say a small vaccine manufacturer, research and development institution where they're researching various risk group 2 pulmonary bacterial infections that all have a very similar risk profile, that might make sense as one risk assessment document. But say you're on the other side, we're a gene editing company working on multiple modalities of viral vectors, so adenovirus, lentivirus, and a novel one at the same time. It might make sense to have three separate risk assessments as those have different risk profiles overall.
Sure. And how invasive would a biosafety risk assessment, and any of these recommendations be? Will this be fully disrupting operations, need to plan around it, or is this something that can be done relatively uninvasively?
Uh, if there's obviously safety considerations in place, then
Sure.
and unsafe work practices, that might be a stop of operations. But I have rarely seen that in my career. Most of the time, as you said, this is a very non-invasive process. We can do a lot of documentation review, away from folks, make sure that we're not disrupting operations, and then to have selective interviews with folks for certain onboarding aspects. But a lot of it is the day-to-day operation. So it's not like they're going to stop work. It's just we're going to prescribe actions to improve the work.
Mhm. Okay, that makes sense. Someone here was also just interested in if you could explain kind of like the difference or the overlap between biosafety cabinets and biosafety itself as a service, because they have, it seems like they have BSCs on their site, but they're not quite sure how that would fit into their larger biosafety program.
Sure. So, I mean, a biosafety cabinet is a tool that we utilize within a biosafety program. So, it's our primary means of an engineering control, so how we contain the work within the site. So, our biosafety consulting services, so we are able to utilize those as part of our program, but Triumvirate also offers a biosafety cabinet certification testing requirements. So it's a separate group from our EHS consulting group, but we do coordinate together those two service lines to keep a facility compliant. But as I said, as a tool, it's our primary means of an engineering control.
Sure, that's interesting to note the tools and the controls versus the service. So someone here is also asking, are certain types of BSCs required in a vivarium? Does it have to be type 2B2, or will type 2A2 suffice?
Uh, so that all goes back to the risk assessment and what you're
Sure.
working with in your facility. So if you are doing, say, a full body transfusion of an animal, so post-mortem doing a formaldehyde fixation of it, because of the risks from those gases, it would make sense to do a class 2 B2 cabinet. So that's a biosafety cabinet that's hard ducted, functionally a BSC fume hood hybrid. So you're dealing with any biological risks from the biosafety cabinet and the HEPA filter mechanism, and then from the paraformaldehyde or the formalin fumes, there's the fume hood aspect and the gas capture and evacuation. So for most facilities, an A2 might suffice, but if you're doing something with chemical hazards or other volatile processes, a B2 might make sense.
Depends kind of on the facility and what's going on there. Got it.
Exactly. Always go back
Cool.
to the risk assessment.
Nice. And actually, it's funny you mentioned that, because we do have a couple questions coming in about risk assessments. What key details do you collect for risk assessments? What's your biggest challenge when completing these risk assessments?
I covered some of the key assessment items on one of my first slides, but we look at what agents are in use, what the risks are of the agent if someone were to be exposed to it, and then the routes of exposure. So if it's a aerosol transmitted agent, that obviously we're looking at how to mitigate the aerosol risks. But if it's just something that gets through only percutaneous injuries or as a bloodborne pathogen, less concerns about aerosols. So we're looking at the agent, the processes in place, and how to set up our program to mitigate the risks from those processes.
That's great. And then there was another question, just a clarification on the question I asked you before about, in general, do you see risk assessments that are performed on specific agents, or generally are people looking at their whole facility wide? I know you kind of touched on this, but someone just wanted some clarity again on that one.
Yep. So generally, for if it's a biotechnology or life sciences company with a small footprint, it's typically one overarching risk assessment. But for larger sites with multiple large scale projects, those will be segregated risk assessments. So it just depends on the size of the facility
Great.
to answer your
Okay.
question.
Yeah, that's good. I hope that was enough clarification. Whoever asked that question, please let us know if you need more information later on. We can send some more. You did also mention dual use research of concern, the DIRC, in the presentation. So, how can an institution incorporate a framework of DIRC into biosafety committee process?
Mm, that's a great question. So the ultimate answer is we don't know how the new DIRC policy is going to be worded and what those requirements are going to be. But from the binary policy and best practices, my recommendation would be to update your biosafety manual and IBC charter to include the DIRC and PEP processes. And there's two modalities you can either take from there. Either your entire IBC could be the DIRC point of review, so that could be a overlap committee, or you could have a subcommittee within your IBC to actually be the DIRC point of contact. So it's not bogging down the entire IBC operations to do these levels of review, since they're not entire. The Venn diagram is not always 100% overlap for those kind of work.
Cool. Yeah. And someone here is just wants a little bit of clarity too on that idea of an IDC. Is that something that someone will have internally, that will have external members in it, or can you just go in a little more detail for someone that may not know what that IBC is?
Sure. So the institutional biosafety committee, so those are typically made up of people within the institution. So the recombinant DNA experts, as well as the any containment experts for specialty work like animals or plants.
Mhm.
So those are all typically internal to the company. And then some large institutions might have biosafety professionals on staff. So those would be internal. But often for the clients we support, we are the external biosafety support. So those biosafety professionals and officers can be an external voting member. And then there are non-affiliated community members that often sit on these committees. So it's a requirement if you have NH funding that someone from your community who does not work at the institution sit on those committees to review the work overall.
Mm. And do all viral vectors, a bit of a tongue twister, required to go through IBC review?
Uh, Ultimately, yes. So they're all recombinant work. So depending on how they were ultimately generated and from what source agent, depends on the level of review. So lentiviruses need full committee review prior to work beginning, and then certain AAVs, because they're risk group one, work can begin simultaneously with IBC notification. A little nuance, but ultimately, yes, all viral vectors do need IBC approval.
Sure, maybe I should provide a little more context about that for the person asking this question too. Someone here is asking about a herpes B program with macaque samples and sharps. So, should a full herpes B program be established with a local hospital for sampling, sending to the CEDC, and providing post-exposure prophylaxis, or what would be the recommendation for a lab working with macaques?
If they're actually working with full live colonies of macaques, they would obviously need a full scale occupational health program and do baseline monitoring of the animals themselves. But then all employees would need to be in an occupational health program for herpes B, MTB, or any other zoonoses that the monkeys might have. But if a facility is working downstream of those and just receiving tissues from those colonies for doing their preclinical studies, then a full herpes B program I would not recommend, but they would need potentially some BSL-2 plus enhancements that we talked about. So some of that would include occupational health interface for mostly post-exposure responses, but depending on the animals in question, there may be some pre-exposure monitoring that may need to occur.
That's great. Yeah, thanks for that information. Here's a kind of a question more about general. It's general and it's specific. So someone's asking what the HEPA testing requirements and how long people should maintain records of these HEPA tests.
Sure. So for biosafety cabinets, the HEPA filters in those should be certified annually. And then to comply with OSHA requirements for that, those records should be kept for at least 3 years.
Awesome. People are also very interested in biohazard waste and waste regulation disposal. What are some of the regulations as it pertains to the storage of biohazard waste? Specifically, someone here is asking about hallway storage for regularly accumulated large amounts of biohazard waste that don't have enough storage space.
Interesting. So regulated medical waste or biohazardous waste or special medical waste is going to very much depend on which state the institution is in. So all those terms I just listed, that's what some states call those, and it's all kind of differ by each state. So do you know what state this person happens to be in?
They did not say. If you're able to, please let us know what state you're coming in from, but not at the moment.
Okay. So my initial guess would be unlikely no, depending on the state. So most regulated medical waste storage areas need to have restricted access. They need to not be in a public space where a lot of foot traffic is going through. And it needs to make sure that you're not storing it for typically 30 to 60 days after the initial generation. So if you're accumulating the material, depending on the state, you can't keep it on site for too long. So that overflow in the hallway is a little concerning on that aspect.
Sure. Yeah, that makes perfect sense. You wouldn't want someone interacting with that as they walk through. We're also getting a lot of questions here about training. So does Triumvirate Environmental come in and train internal teams to do risk assessments, or are they training for something different?
So we can offer any levels of that type of training. So we can come in and functionally train you, work with you to understand the risk assessment process and how to understand biosafety as a workflow and give you that go forward knowledge. Or we can come in and do trainings for the facility operations and actual research and production staff to do the safe work with the biologics safely. So we can either do your biosafety training or train your biosafety staff.
Sure. And you had mentioned earlier, you mentioned bloodborne pathogens training. Are there other similar, like shorter form or more accessible trainings that employees should be doing when there's an assigned biosafety level off the top of your head?
So they should have a basic biosafety training in addition to the bloodborne pathogens one, especially if they're doing BSL-2 work. And then everything will, from that point on, is based on the risk assessment. So if they're doing BSL-2 plus work, specific training on lentiviruses or the NHP material, and what those BSL-2 plus enhancements mean. And then obviously for BSL-3 training or any agent-specific trainings, they'll need something specific. Less short form for those since those are a little more involved, but at least digestible in those aspects.
Sure. Yeah, that makes sense. And someone here is asking about training for IBC members. So is there certain training that is required to be a member of an IBC or certain degrees that are needed to qualify their expertise in NIH guidelines?
So as far for yes and no. So if you're the person is filling their role on the committee as a containment expert or recombinant DNA expert, there at least needs to be demonstration of they have expertise in those fields. For general IBC members, there is requirements within the NIH guidelines that they be trained on the role of the IBC, what their role means, and how the IBC works. So we can tailor training to each IBC, to the IBC members, so they understand what the scope of work is, how to conduct a risk assessment on behalf of their institution, and how to work with their own IBC.
Great. And how long would any of relevant training that we've talked about for IBC members or internal trainings, how long does the training last? When does recertification become necessary?
Oh, for new IBC members, that's a typically one-off training, and depending on the complexity of the committee and the institution, anywhere from 15 to 45 minutes.
And then recertification annually, once every 3 years?
So for just conducting an IBC meeting, I wouldn't recommend ongoing renewal training unless there's
Okay.
obviously a lot of turnover within the committee. But for other biosafety training for people working in the labs, those trainings are at least typically annually.
Cool. Okay. Someone here is asking now about cadaveric tissue. Excuse me if I'm mispronouncing that. What are some general recommendations that are needed for a facility working with that tissue?
Yep. So if you're working with cadaver tissues, so that's obviously human source material. So the bloodborne pathogen standard comes into place. So all employees will need to undergo bloodborne pathogen training, be offered the hepatitis B vaccine, and then you'll have to go through what actual processes you're doing with the cadaver tissue. If you're doing dissections on an open table, we'll obviously look at what face and eye protection you'll be wearing, additional PPE. But if it's just small scale tissues that you can handle in a biosafety cabinet, then those obviously should be handled there. But if it typically would recommend a bloodborne pathogen program that looks at PPE administration to mitigate those risks.
Sure, that's so interesting that you mentioned PPE too. So is that a common factor in some of these biosafety programs and risk assessments? We had a service spotlight in January that talked about industrial hygiene. So they covered PPE a lot too there. So maybe can you explain a little bit more the relationship between industrial hygiene and biosafety and PPE and biosafety?
So PPE is always a safety professional's best friend, but it's the
Sure.
last line of defense we like to implement. So whether you're working with a basic chemical reaction or a basic biologic process, you kind of be wearing PPE within the lab. So for basic biosafety like BSL-2, folks are going to be wearing a lab coat, gloves, and typically safety glasses. But when you're looking at industrial hygiene processes, just like when you're doing biosafety, it all goes back to the risk assessment. There's a lot more direct controls within an industrial hygiene because it's based on federal guidelines around particulates and exposure limits. For biosafety, it's less of an 8-hour work duration because if you get exposed to an infectious dose of whatever agent you're working with, it doesn't matter if it was a single instance or over an 8-hour period. So biosafety is more focused on a single instance, whereas industrial hygiene is a large scale focus of the entire process. But not to say biosafety isn't focused on looking at start to finish of a safety program.
No, that's so interesting. I really appreciate being able to know the difference between those two. I'm sure a lot of people do too. Here's an interesting one. Like you talked about BSL-2, BSL-3, all these different biosafety levels. What about BSL-1? Are there any specific concerns for that, or is that just a lab that's operating as normal? Can you explain BSL-1 a little bit more?
So BSL-1, it's a basic lab. So most of the equipment and facilities you're going to have in that, you're going to have a sink for hand washing. You're going to have surfaces that are easily decontaminatable. If you spill anything, you're going to throw the waste away and make sure it's not going to be released into the environment, even though it's all BSL-1 and otherwise safe. These are things you can typically do in your kitchen sink. And just like I would recommend it in any kitchen, whether it's professional or home, the basic fundamentals of hygiene are the biggest thing a biosafety professional. So that's why people who've been at BSL-1 still need to go through training to understand how they're supposed to act in the lab, what PPE they're wearing, that they're supposed to wash their hands, how to do that in the facility, and then how to get rid of their waste. Even if it is non-hazardous, it still has to be or approached intentionally.
Yeah, that's interesting that you mentioned waste. So, how does Triumvirate Environmental go about disposing of biological waste or biologically contaminated waste, I would say?
Yeah, so for our BSL-1 and BSL-2 clients, we have a lot of modalities, and BSL-3 I can talk about as well. For our BSL-1 and 2 clients, most of them are treating their liquid waste on site, so either through bleach or some other EPA-approved compound. All of that actually is approved and reviewed typically by the site's IBC. So that's something we can support is reviewing what your waste is, looking at on-site treatment options, whether it's through chemical treatment or if you have an on-site autoclave. There are state regulations around autoclaving on site if you're doing that, so we can help you comply with those, make sure you have a trustworthy autoclave program, and make sure you're complying with any validation and spore requirements for that. However, generating that waste and you don't have a way to deal with your solid waste or any liquid waste on site, Triumvirate is a disposal partner you can work with. So we will take your liquid waste or your solid waste, most germanely, take it to one of our facilities, and typically we will autoclave that on site and recycle it afterward into our various waste streams.
Great. Oh, that's awesome. And someone here is asking a little bit more about the biosafety level. If this person in particular, it seems like they're taking over a lab or they're opening a brand new lab. How do they go about knowing if they need a assigned biosafety level, and then how do they actually go about getting that like biosafety level distinction?
So again, it all comes back to the risk assessment. So you'd look at
Mhm.
what agents you're working with on site, if you have an IBC, or if it's dictated by that. So if a biosafety level isn't something you fall into as a facility, like just happenstance, you're going to probably well understand that the biological agent going into that, either the design process or when you're looking to lease the space. But as far as designation from once you understand you're at a biosafety level, if you're looking at BSL-1, 2, or 2 plus or 3, that's up to the risk assessment and the agents you're working with.
Sure. And kind of to this point again, I know we keep coming back to this idea of a risk assessment. What are some of the telltale signs that someone may need a reassessment or to do an assessment for the first time, a biosafety assessment?
Yep. So if you're doing an NIH compliant work with recombinant or synthetic materials, all that does need to be re-reviewed by the IBC at least every 3 years. And depending on some processes, they'll often wrap that into an annual review. But that's the bare minimum as far as doing a reevaluation or a reassessment. If there's any new work that's occurring, new processes like new equipment that you're bringing in, if you're doing say flow flow cytometry and sorting, where you hadn't been doing that before, that may warrant a risk assessment update and a representation to the IBC to make sure it's still a compliant program. For new agents, so that's an obvious one. If someone's bringing something new to your institution or facility, that should be reviewed as well.
So here's someone that says that they're hiring a new vendor to bring in some offsite external equipment from a manufacturer. Will those will the biosafety programs of those vendors impact their internal biosafety program or general lab safety program?
Oh, interesting. So if you're bringing a contractor in from a third party to either service your equipment or bring something in new, often this is wrapped into a larger vendor safety program and not maybe just a biosafety specific question. So oftentimes I would not have a vendor work on a piece of equipment if it wasn't decontaminated before they were working there. But if for whatever reason they had to be, we want them either to make sure they are equivalently trained, either by the same on-site staff and training that those people would go through, or some confirmation that that vendor had received appropriate biosafety and bloodborne pathogen training.
Yeah, that makes sense. Just checking, crossing all your bases and everything. Um,
Exactly.
Yeah, that's great. Does biosafety work continue after appropriate mitigation measures have been implemented? Like, for example, is there any monitoring or reporting required on some sort of regular or other sort of basis?
It's going to very much depend on your facility. So if you're an NIH funded institution, you have a registered IBC, any releases or accidents with materials should be reported either within a couple of weeks for a couple of days, depending if it's a BSL-3 organism, or within 30 to 60 days, depending on the other nature of it. There's going to be local or state public health departments that may need to be notified of certain releases or incidents, depending on the agent that you're working with. So classic biosafety answer on that one of it depends.
And is that release kind of data tied in at all to the TRI, like toxic release inventory reporting that? No? Okay.
No,
Interesting.
so biologics and those chemical kind of toxins or leachers are pretty distinct in that aspect. So most legislation, if they refer to a toxin, they're not referring to a biological toxin outside of these new DIRC or PEP policies we're talking about.
Okay. Someone here is saying that they have a current site that has a biosafety program. They're opening a new facility that's doing the same type of work in the same state. Can they just copy what they did on their original site onto their new site, or do they need a separate biosafety risk assessment and program?
So if the work is identical, the biosafety manual will probably be virtually identical, but it still does need to be site specific, and there needs to be an evaluation to make sure that everything is aligned. So the risk assessment ultimately would lead back, make sure that you're aligning with that your old facility is the same as your new facility. However, if there's maybe HVAC changes or engineering controls or workflow processes that might differ from one facility to another, that should obviously be taken into consideration within the biosafety manual or associated risk assessments.
Sure. Yeah, and we've been talking a little bit about HVAC and we've been talking about HEPA. What are some examples of common risk mitigation strategies that you may implement during a biosafety program update?
Oh, as far as an HVAC controllers or HEPA filtration goes?
I mean, kind of like beyond that. Like, are there any common, like, low hanging fruit or quick steps that someone could start to have a safer operation in terms of
Oh, I mean,
biosafety?
Uh, again, it very much depends, but a classic answer is just improved training. So oftentimes these facilities have either outdated or very basic non-site specific training. So the best thing I could recommend is something more engaging and then site specific that personnel or the folks will actually listen to and stick better.
Sure. I think we have time for a couple more questions. I am getting a few about this biosafety manual that you've mentioned.
Mhm.
The first question is kind of around who is helping update and maintain that? Is that the IBC? Is that an external consultant like Triumvirate? Is that internal employees when they notice issues? Who's kind of helping maintain or update that biosafety manual?
Yep. So it very much depends on the facility. So for larger institutions with a dedicated EHS group, that's typically the folks who hold that responsibility. But for many of our clients that we're working with, we're either partners within that process or for we are the initial generator and then the first stop for the annual revision of that biosafety manual or the bloodborne pathogen exposure control plan. So many times for larger institutions with more complex work, the IBC will review and approve those biosafety manuals, but it does depend on the size and scope.
Yeah, and there's someone here that's asking, they know that there are certain emergency response procedures and, like the chemical information that needs to be readily available to employees. Does the same apply to a biosafety manual? Does it have to be kept in a common space, or is it more of just access it when you need it?
So just like a chemical hygiene plan, the biosafety manual and bloodborne pathogen exposure control plans do need to be easily and readily available to employees. So for most sites, they're going to do that, comply the same way they would with their chemical hygiene plan, either printing it off and having that available or having an easily accessible online repository.
Sure. Okay. Yeah, that makes perfect sense. Someone here is asking again about our specific services here. So, are we able to do one-off projects with clients, or does it have to be full management of the biosafety program?
Oh no, it can absolutely be a single one-off project where we come in for a couple hours and have a dedicated tasks, all the way to full program management, to where we have dedicated 1 or 2 day a week a resource dedicated to that client.
And does that include any like emergency response that may be necessary?
Absolutely, yeah. So we can have an ad hoc agreement for those kind of services.
Awesome. Here's just one more quick question I want to get in before we jump. Does biosafety risk assessments and general programs, do those consider wastewater as well as biological materials, or is it only one or the other?
Uh, well, so if live waste is being discharged, that's obviously something we consider within the wastewater. So depending on the state and municipality, some of them do allow discharge, like the state of Massachusetts. If you're working with risk group 1 material that's still recombinant, your IBC can review that and confirm that it would be not a risk to public health, but some other states don't allow that. So it'll very much depend on which state you're in and if you need to fully inactivate your waste before discharge or have consideration for what you're actually discharging.
Seems like the moral of the story is it depends. Let us know. We're going to help you out.
Exactly.
Awesome. Well, that's all the time we have for today. I just want to say thank you so much, Brandon. You answered so many questions and it was incredibly informative. So we really appreciate that. And a huge thank you to everyone who's attended today as well. As I mentioned earlier, you will receive an email with a copy of the presentation and the slide deck. Feel free to reach out if you have any further questions for us. After the event, you'll also be directed to a short survey about your experience today. A link to the short survey will also be included in the follow-up email. We'd greatly appreciate your feedback on this Service Spotlight format and the event overall, including any ideas you have for future topics. Thank you so much for your participation today. We look forward to seeing you at future upcoming events. Take care.